Good morning and good afternoon, and welcome to TransCelerate's Demystifying Quality Tolerance Limits webinar. We're happy to conduct this webinar, the second of two webinars, and are happy that you were able to join us. On the call today, we have Lucas Berzarski, Dagmar Gortz, Ruma Bagat, all three are team members and will share their experience in going through and developing a QTL framework that's available to anyone following this call. My name is Leslie Ware, and I'm the Program Manager for Interpretation of Guidances and Regulations. QTL runs underneath that. Next slide, please. Let's go through just a few logistics for the webinar. Everyone will be muted for this call except for the presenters. You have an option to reduce the GoToWebinar control panel. There is an orange button you'll see in the upper left-hand corner. Simply click on that to reduce that. Also, during this session, you'll have the opportunity to ask questions. If you look in the webinar panel, you'll see a gray bar that says questions. Please feel free to submit those questions, and when we reach the question and answer portion of the webinar, we'll try to select and respond to as many of those as possible. Be sure that you click the send after you put your question in the box to make sure that it goes to the panelist. This is being recorded, so if for any reason you are uncomfortable with that, even though you're on mute, please just send a note using the questions panel. Next slide, please. To make this an interactive webinar, what we'd like to do is some live polling, and we're going to do a little test here just to make sure everyone can know how the polling works. It's all voluntary and anonymous. So you don't have to respond if you don't want to. But what we'll do when we reach a few questions or polls is we'll launch that poll. And so let's test it right now. We're going to launch the poll, and I'm going to ask you to select your favorite color. All right, looks like everybody's knows and has figured out how to do the poll. That's great. Let's see, I'm just watching the numbers here. As with most of the webinars, blue is the winner. I think we can close out the poll now and share the results. So 55% of you say blue is your favorite color. So well done, everyone. Let's go on to the next slide. Just a few ground rules. Again, participation with using anything that TransCelerate puts out is voluntary, and when we do questions and you submit questions to us, just a couple of rules around that. You don't have to identify what company you work for. We do ask that you avoid talking about vendors, sites, CROs, any issues you have along those lines. We won't be able to respond to any of those. And then we also suggest that in your questions that you avoid any sort of long-term development plans for your company and anything related to cost. I think we're ready now to move into our agenda for the webinar. We're going to start with just a very quick overview of TransCelerate. I'm sure many of you know who TransCelerate is. But we'll just touch on a few things there before we actually go into the objectives and methodology this team used for this initiative. We'll talk about what is a quality tolerance limit. We'll propose a process that could be followed, and then we'll go through just some general considerations. We'll end up with a Q&A session at the end. But before we get started, let's do two more polls. Please rate your company's current understanding of QTLs as it pertains to clinical development. So everyone go ahead and if you'd like, provide that background. So I'm watching everybody put their responses in. This is fantastic. We're seeing somewhere, some understanding, good understanding. Some folks have really limited understanding at this point, and what we're hoping to do is increase that understanding. So I think we can close out the poll now and see how you responded. Good. All right, so I'd say about 50% of you, or over 50% of you, are saying that you have some understanding, limited understanding. Let's hope that as a result of this webinar, we're able to move that up a little bit and help you feel a little bit more confident and comfortable with what a QTL is. Let's do one more poll before we start off. What's the greatest challenge your company has in adopting QTLs? And you can check all that apply. All right, great. Got about 30% of you have responded, having to give that a little bit of thought and read through. What are the specific QTL parameters, setting thresholds, what is, coming up with a process, and then deciding how to report out. Great. We've got about 70% of you have voted, so I think we can close out the poll and see what you thought. All right, so about 45% of you said you're having challenges with all of these particular areas. So that's really great, and I hope, again, that we're going to be able to hit some of these areas and again, help provide some insight for you. Let's go on to the next slide. I'm just going to give you a quick overview of TransCelerate. Next slide, please. As probably many of you know, TransCelerate is a not-for-profit entity, and we work in we want to foster a collaboration. We have a shared vision to improve the health of people around the world by accelerating and simplifying R&D for innovative new therapies. Next slide, please. We're actually made up of about 20 pharmaceutical biotech companies, but our membership, even though the individuals that work on this all primarily come from all of these companies. We actually only have about 30 employees with employees of TransCelerate itself. Next slide, please. This next slide reflects all of the various initiatives that TransCelerate has worked on or is working on. This particular initiative is under an area we call sponsor efficiencies, and in this particular work stream, we strive to deal with some of the ambiguity that comes out of certain regulations and guidances. I'm going to go ahead and turn the webinar over to Ruma, and so next slide, to talk about the objectives and methodology.
Thank you, Leslie. Thank you, Leslie. I'm Ruma Bhagat. I'm a senior science leader in the Health Equity Population Science team at Roche, and I will present to you the initiative objectives and the methodology. Next slide, please. The QTL complements the other TransCelerate risk-based management initiatives, the risk-based monitoring, quality management systems, the protocol deviations, and the quality tolerance limits. They all form part of the risk-based approach to quality management at a clinical trial. Next slide, please. The objective was to propose approaches for end-to-end process for implementation of QTLs as defined in section 5.04 and 5.07 of the ICH E6R2, including considerations for setting thresholds. The impact was to improve the systematic quality of clinical research through a framework on implementation of QTLs in a way that would facilitate compliance, efficiency, minimize confusion, and avoid duplication of efforts. Next slide, please. Here are the deliverables overview. As I mentioned, the QTL deliverables are intended to aid the industry's ability to improve the quality of clinical research by facilitating the implementation of QTLs in a way that meets health authority expectation, is efficient, minimize confusion, and avoids duplication of effort. The QTL framework and FAQs is one of the main deliverables of our workstream. These documents intend to provide definition of the term QTL, a high-level process map, and steps for defining, monitoring, and reporting of QTLs, examples of QTL parameters in the QTL library, the considerations for setting thresholds, and answer to some common questions. The QTL plan template is a trial-level template sponsors can leverage to help develop and determine the QTL monitoring plan, how the QTLs will be managed through the life of the study, changes to QTLs throughout the trial, and justification of these changes. The QTL manuscript. So we now have a publication in the DIATS journal about the QTL framework and the implementation of QTLs. This is a peer-reviewed publication that details the proposed definition of QTLs, the high-level process map, and again, some examples. We also have a CSR and CPT template text. These are the text which is proposed for the model documents to be included by study teams. Coming soon is publication 2 that shares and speaks to the QTL historical benchmarking data. We hope that this will be available in Q1 of next year. Next slide, please. The QTL framework components, one of the most important deliverables is the framework. This contains the risk-based quality management components, a step-by-step details of the end-to-end process for QTL implementation of define, monitor, and report, and an appendix which contains the QTL library as examples of QTLs that can be set up for the study. Next slide, please. I'm now going to hand it over to Dagmar to give us more details on what is a QTL.
All right. Thank you so much, Ruma. Good morning, good afternoon, and good evening, everybody. So my name is Dagmar Goertz. I'm Director, External Engagement and Business Insights at J&J, and I'm also a member of the IGR QTL team. So what is a QTL? What is a quality tolerance limit? Before we take a look at the description, I invite you to participate in another poll, please. So, Fran, would you mind? No, thank you so much. So what do you associate with QTLs? Is it GMP? Is it GCP? Is it risk control and reporting? Is it patient safety? Or is it all of the above? Right. 66% go for all of the above. We have almost 70% who voted. Okay, almost 80% of you voted, so maybe you want to close the poll and take a look at the results. And the majority of you said all of the above. So next slide, please. Thank you. So all of the above is what the majority of you responded, and that is right. So let's take a step back and look at some history and background information. Historically, QTLs have been established in product manufacturing environments with the purpose of maintaining acceptable product quality via a reproducible manufacturing process. In a product manufacturing environment, a QTL is established to trigger a decision on the acceptance or non-acceptance of the manufactured products. Batches found to be outside of these limits may be discarded, ensuring that poor quality products do not reach the next phase of assembly or even the consumer. New products would be manufactured to replace the discarded ones. This QTL framework from the product manufacturing environment cannot simply be applied to clinical research and development environment. So hence, implementing the QTL process as part of GCP requires adaptation. In clinical research, poor quality data can result in the inability to use some of the trial data, leading to the inability to draw conclusions. It is therefore important to implement proactive measures to ensure data quality by leveraging historical knowledge and fostering quality by design, also known as QBD. ICH E6R2 introduced the concept of QTLs to identify systematic issues that can impact subject safety and reliability of trial results, and ensure that important deviations from the predefined quality tolerance limits and associated remedial actions taken are reported in the clinical study report, CSR. Next slide, please. So what are the underlying regulatory expectations? The ICH E6R2 guidelines were published in November 2016 and adopted by various health authorities in 2017. Section 5.0 quality management was added to the guidelines with the introduction of risk-based approaches to quality management for clinical development, including the concept of quality tolerance limits. With the introduction of ICH E6R2, sponsors have experienced challenges with interpreting and implementing QTLs that meet health authority expectations. So let's take a quick look at the relevant sections and information in ICH E6R2. The section on risk control says, the sponsor should decide which risks to reduce and/or which risks to accept. Predefined quality tolerance limits should be established. Detection of deviations from the predefined quality tolerance limits should trigger an evaluation to determine if action is needed. In the section on risk reporting, it says the sponsor should describe the quality management approach implemented in the trial and summarize important deviations from the predefined quality tolerance limits and remedial actions taken in the CSR. Next slide, please. So what is a QTL? Now that we've reviewed some QTL history, background, and the underlying regulatory expectations, I'd like to describe what a QTL is in the context of GCP clinical research and clinical studies. What you see here is a very, very simplistic illustration of the key elements of a QTL. Parameter plus threshold makes a QTL. A QTL is a level, threshold, or value associated with a parameter which is critical to quality. QTLs are set for risks identified at the trial level. A deviation from a threshold during the conduct of the trial may indicate a systematic issue that could impact participant safety or reliability of trial results. Next slide, please. So as we just saw, key elements of the QTL are the parameter and the threshold. But what is a parameter? What is a threshold in the context of QTL? A parameter is a trial variable to be assessed at trial level. These variables could be related to the critical to quality factors of the trial. When used in the context of a QTL, parameters are measures that detect potential issues that could be mitigated. Examples of parameters are percentage or number of study participants randomized who do not meet inclusion-exclusion criteria, or percentage or number of study participants with premature discontinuation of study drug. Thresholds associated with parameters are designed to detect potential issues that could be mitigated. If a threshold is reached or exceeded, an appropriate action is taken. Thresholds are defined at trial level. Early action thresholds, that means secondary limits, could be specified for QTL parameters to provide study teams with early opportunities to mitigate risk to participant safety or reliability of trial results and avoid a QTL deviation. This added limit allows study teams to intervene before an important deviation from QTL is observed. Now that we've described the elements of a QTL, I'd like to hand over to Lukasz, who will provide insights into the QTL process. Thank you.
Thank you very much, Dagmar. My name is Lukasz Bojarski. I'm heading up the centralized monitoring team at AstraZeneca, and I'm the member of the IGR QTL team. And it's my pleasure to talk you through the QTL process today. Next slide, please, Fran. What I'm going to present today is high-level overview of the process. I will mention key elements of the process, but if you're interested in a detailed description of each process step and looking at more flowcharts describing the process, please refer to the QTL framework. That is one of the tools that were mentioned by Ruma and is available in the TransCelerate website. Next slide, please. The QTL process is divided into 3 stages. The first stage is to define QTL. The second stage is to monitor QTLs. And the third stage is to report important deviations from QTLs in clinical study report. Continuous improvement principles should be applied to the entire process, but there are some elements of the process for which lessons learned from historical and past studies are particularly relevant. For instance, the definition of a threshold for QTL, or agreement and in implementation of corrective and preventive actions that already turned out to be the most effective in the past studies. And the process is really cross-functional, so it of course requires insights from statisticians, medics, development operations experts, centralized monitoring, data managers, and so on. Next slide, please. Let's start with the review of process steps within each stage of the QTL process. The first stage is to define QTL. It should start when the study protocol is still in draft, and important inputs to this process include clinical development plan and draft protocol or study synopsis. And since quality tolerance limits are one of risk controls that belong to the quality management system, the first step in this process is to perform risk assessment in order to identify critical to quality factors and critical data and processes, and applying the quality by design principles. This step, risks associated with critical to quality factors need to be identified as well. Next slide, please. The second step in this stage is to define QTL, which includes definition of a parameter, so agreement on a study level parameter that will be monitored with a QTL, provision of a description of that parameter, as well as definition of thresholds and provision of their justification. In order to come up with meaningful thresholds, one needs to consider statistical characteristics of the study, medical characteristics of the study, historical data in terms of the historically observed issues, how they accumulated in similar studies. But there are also other elements that need to be considered in this step, which includes availability of data sources. We need to be able to come up with QTLs that will be possible to be monitored. So data informing the QTL reviews have to be in place. And it's also worth considering if defined QTLs will be actionable, what actions can be taken if a deviation is observed. Next slide, please. In the third step in the definition phase, the QTL monitoring plan should be developed that would capture the time frame of QTL reviews or checks of study performance against predefined QTLs. The time frame consists both of the start date or the start time for QTL monitoring and frequency of QTL reviews. It should also contain information on data sources for QTL monitoring, and if algorithms or any sort of analytics is going to be used to monitor study performance against QTLs, the programming may start in this phase. And this phase should be finalized at the time when the study protocol is finalized and before first patient in. And this is the moment in which the definition phase concludes. If we go to the next slide, please, then we are actually entering the monitoring phase. And the input to the monitoring phase is, of course, QTL monitoring plan that I mentioned in the previous slide. The first step in this stage is to finalize setup of a QTL report, so the analytics that will be used for QTL monitoring. This involves programming and configuration, data mapping activities, and of course validation of the report. Next slide, please. Then the report needs to be run in line with the time frame, start time, and frequency specified in the QTL monitoring plan. And once the report is run, of course, the outcome or the output of the analysis needs to be assessed. And at this stage, there are two possible scenarios. In scenario number one, the deviation is not identified. The deviation from QTL is not identified. So basically, no action is required, and the report needs to be run again at the frequency specified in the QTL monitoring plan. The teams may also consider performing trend analysis, even if QTL deviation is not observed, to to gain insights into the trajectory of the accumulation of issues, to think about that is it likely that the deviation is to be observed at the time of the next check, and some preventive actions or early preventive actions may be considered at this stage as well. Now, in the scenario number 2, when the deviation is identified, and Fran, if you can move to the next slide, please. One step back, please. Yeah, thank you. So the deviation is identified, needs to be investigated. The root cause analysis has to be performed to identify causes that underlie the observed deviation, and the importance of the deviation needs to be assessed, keeping in mind that deviations that may impact patient safety and integrity of trial results should be assessed as important. Corrective or preventive actions or both needs to be agreed and implemented at this stage. Importantly, during the monitoring phase, teams have also the possibility to of a revision of predefined QTLs. It may happen that during the initial discussion on QTL definition, some factors may not be taken into account when deciding on the threshold, and they may become more evident during the study monitoring or the study conduct phase, and some risks may be emerging in that phase. So they may be taken into consideration in the study conduct phase, and QTLs may be revised. However, it should happen in the extraordinary situations, and always a good justification for doing that has to be provided. And the process is iterative, of course, so the report is run multiple times during study conduct, and whenever the deviation is observed, the root cause analysis has to be performed, and the importance of the deviation needs to be assessed. Next slide, please. So the monitoring stage, it concludes at the time of database lock, and it's also the time when the reporting phase starts. It should start with generation of a QTL summary report that will list all deviations from defined coordinate limits, as well as actions taken to address these deviations. Next slide, please. In the next step, the final review of all deviations should happen, and the final assessment of importance should take place. It may be the case that some deviations that were initially categorized as important may be classified as non-important from the perspective of the end of the study, and vice versa. These which were first recognized as non-important, the classification may be changed into important QTL deviations at the end of the study. And the final step in the reporting phase is inclusion and summarizing of important QTL deviations, as well as actions taken to mitigate these deviations and root causes underlying these deviations in the clinical study report. And it almost concludes my part, but I also have a poll. So if you can go to the next slide, please. Right, so the question for the audience is, when defining a QTL, the following factors needs to be taken into account: historical data, statistical design of a trial, availability of source data for QTL monitoring. Please check all that apply. It's time to vote now. I can see that almost 60% of audience has already voted. Let's keep it for a moment. Open. Okay, I think we are at 70% of votes, so let's take a look at the results. Right, so the vast majority of audience responded that all of the above had to be taken into consideration, and this is the right answer, as both historical data, statistical design of a trial, but availability of source data for QTL monitoring are important when defining QTLs. And with that, I would like to hand over to Ruma, who is going to speak about general considerations for quality trans limits.
Thank you, Lucas. Next slide, please. So the historical data when setting threshold analysis, the historical analysis can be useful to consider in establishing QTLs. Consider the following factors in selecting these historical studies: the therapeutic and disease area, the trial phase, the participant population, example, the stage of disease, age, gender, whether or not historical data is recent enough to represent the current processes, and relativeness to standard of care. Ensuring an appropriately relevant comparison between the trial data and historical data is important to set meaningful thresholds. Next slide, please. The identification of systematic errors or data bias in completed trials may be difficult. Below are some tactics that can help us to avoid this. Excluding any significantly outlying trials. The median may be a more relevant data as inherently eliminating outliers. Investigating previous CSRs to learn from past systematic issues, and leveraging the most recent trials as they may be a better representation of the current practices. Next slide, please. Special consideration trials. Sponsors can determine the applicability of specific QTLs to early phase trials based on the clinical development plan for the molecule. The ICH E6R2 guidelines should be followed when generating clinical trial data intended to be submitted to regulatory authorities. The ICH E6R2 quality management section mentions that the methods used to assure and control the quality of the trial should be proportionate to the risks inherent to the trial and importance of the information that is collected. Next slide, please. Here are some criteria that can be considered to assess the applicability of and the number of QTLs as quality measures to early phase trials. The number of participants in the study. A certain number of participants are needed to avoid low statistical power or false QTL signals. The number of sites that are needed to avoid low statistical power or skewed QTL reports. The duration of the study. An adequate duration of the trial is a consideration for implementing QTLs and any remedial methods. If the duration is very small, there may not be enough time to be able to implement the QTLs and have any remedial actions. The recruitment rate. The trials with a very low recruitment rate may lead to false positive, false signals and low statistical power. And trials with a very high recruitment rate may not offer us the ability to set up the QTLs and remediate any QTL signals, as enrollment may be over very quickly. The trial design. Some trial designs may be challenging to implement QTLs. Example, dose escalating cohorts due to a small number of patients in each cohort. And the trial population, heterogeneous versus homogeneous trial population. Next slide, please. The number of QTLs. The number of parameters used in a trial should reflect the number of critical to quality factors to which risks are foreseen in execution in the execution phase of the trial. The execution-related critical to quality factors are primary objectives, key safety objectives, patient eligibility, and investigational product exposure. 1 to 5 parameters may be appropriate depending on the risks identified in the trial. Some parameters may include more than one CTQ factors. Example, percentage of patients with eligibility violation can be effective of both quality of the patient population recruited and the safety of the participants. Based on the trial design, some CTQ factors may not be at risks of systematic issues. Example, in eligibility of patients for a rollover trial, eligibility may not be a risk for these studies. Next slide, please. Use of trial-specific QTLs. QTLs must be based on the medical and statistical characteristics of the study and need to be study-specific. The thresholds need to be calculated for each study. The pros for establishing the trial-specific QTLs are that they are most likely to offer more scientific value than generic parameters. It may ease the adoption of QTLs by trial teams by being directly correlated to their trial protocols, facilitating a change management in the introduction of QTLs. The cons of this approach is that it may increase operational complexity of QTL monitoring. New parameters will generate a need for additional programming and validation and increase resource demand. A middle ground solution may sometimes work by establishing therapeutic area-specific parameters. For example, in many oncology trials, parameters focused on results data quality or completeness may be closely related to the scientific value and applicable to a broad set of trials. Next slide, please. With that, I hand over to Leslie for a summary and our Q&A session. Thank you.
Great. Thank you, Ruma, and our panelists for a very detailed overview of what you've been able to put together. Before we actually go into our Q&A session, I'd like to do a couple of things. First is to share with you where you'll be able to find a wealth of material that this team has produced. All of this is available on the TransCelerate website. We have a framework that goes into much more detail than what we went through today and aligns with what you heard today. We also have an infographic that can be used. It's a short infographic that can be used in your company to explain what a QTL is. We also have a peer-reviewed publication in DIA Therapeutic Innovation Regulatory Science. There's a link on our website to that article. There's a template that you could use for setting your own QTLs within your own company. And we have a series of FAQs. And then along the lines of the relationship we have with the Clinical Content and Reuse Initiative for TransCelerate, you'll find some standard text in both of those templates that you can leverage if you'd like. All this, again, is available on the website. We also, following this call in the next couple of days, will send you a link to this and also the recorded webinar. One last thing before I move off of this is to just share that we do have an upcoming webinar on what we call the TransCelerate Quality Quartet. It's RBM, QMS, protocol deviations, and QTL. You should be able to find access to register that on our website, and we'll also provide a link to this upcoming webinar toward the end of the month in the email that we send out to you following this session. Let's do a quick poll now to see if we were able to perhaps change, maybe hopefully increase a little, at least a little bit, your company's current understanding of QTLs. We're going to answer the same question that we did before, but let's see if we were able to change that. So let's launch that poll. So if you wouldn't mind sort of considering after we've gone through this webinar what you feel your current understanding is. So about 60% of you have voted. I would say that we started out with 14% saying that they had limited to no understanding, and that is 0 right now. So clearly there's been an increase there. So a lot of upward movement in understanding. I think we're at about 70%. So let's go ahead and close that poll and let you see. We've also moved up to I think in the good understanding previously was 32%, so now it's 52%. And the very good understanding previously was 9%, and now that's moved up to 21%. So I'm hoping that you walk away from this feeling that you were able to enhance that understanding. The poll seems to indicate that you did. And then certainly take advantage of the tools that are available to you to continue to enhance your understanding. One more poll we have for you, and that is to ask, how likely are you to go onto the website and leverage the quality tolerance limits framework? Go ahead and launch that poll. And keep in mind, there's a series of assets there for you to leverage beyond just the framework itself. All right, we're moving on up. With those of you that have voted, we're about 62%, 63%. I think we can close out the poll now and see. Great. So pretty much about 95% of you are saying, "Yep, you're gonna go take a look at that and leverage those that framework, the information that's there." So I think at this point, let's move on to our Q&A session with our panelists.